Hello, everybody. My name is Patrick Lange, and I warmly welcome you on the webinar, How to Achieve Compliance in Quality Control of Nitrogen-Containing Compounds. Our panelist today is Dr. Edward Wiedenbeck. Edward, can you show up at that moment? He's a product specialist in Kjeldahl Solutions. He holds PhD in chemistry from the University of Constance in Germany. His pre- and postdoctoral pharmaceutical research projects have been focused on drug delivery and crystallization techniques. Edward joined Biki in 2020 as a product specialist. So the audience is yours. I will just mention that at the end of the presentation, there's going to be Q&A session, which we will lead together.
So hello and welcome everybody to our today's webinar. Also from my side, it's about how to achieve compliance and quality control of nitrogen-containing compounds. And first of all, I'd like to tell you something about the learning goals of this webinar. So we will talk about how to use nitrogen content determination as a precise method in quality testing of any compound. Second, we will talk about the main aspects for achieving compliance to 21 CFR Part 11. And third, in the end, I will show you 3 steps to establish a time-efficient and compliant SOP for quality testing. So, and as already mentioned in the invitation email, there will be a bonus at the end of the webinar. So you can see a real-life case study of how Sanofi has adopted the Kjeldahl method for compliant QC in their laboratories. So the main question is, why should we focus on nitrogen in compounds? And therefore, in order to answer this question, I brought some image from a paper from Journal of Medicinal Chemistry, where they examined almost 2,000 FDA-approved drugs and focused on more than 1,000 unique small molecules, and found out that more than 870 of those do contain at least one nitrogen atom, which means that's approximately 84% of all these drugs that are approved by FDA. And you see that once you have a nitrogen atom in your chemical structure, it is eligible for the Kjeldahl method itself. And now you might ask yourself, what are the specific fields of application in pharmaceutical industry? So Kjeldahl analysis is a primary reference method for nitrogen determination in quality testing of pharmaceutical manufacturing. And the fields of applications include raw materials, nutraceuticals, APIs, excipients, and blood plasma products, for instance, and vaccines. So yeah, actually the field of application is really independent from this side. It's just it has to contain any nitrogen in their chemical structure. So these are all the possible fields of application that you may encounter also for the Kjeldahl method. And maybe it's best now to jump directly to the Kjeldahl method itself. So the Kjeldahl method consists of 3 steps in total. And yeah, let me first talk about the first step, the digestion step. In this step, you basically use your organic compound, of course, with the nitrogen inside. And yeah, you add sulfuric acid, and at a high temperature, you have a digestion reaction. It's like a decomposition reaction. And then you end up with carbon dioxide, water, and ammonium sulfate, which is the key product for further analytical steps. And here you can see how this will look like. So during digestion, you will see some foam formation in your sample tubes, and then you end up with the clear green digestate over here. So there's your sample inside. So, and here we can see a video how this looks in application in real life. So all these sample tubes, they are in a rack, and you place it basically into the digestion unit. And then you close the suction module to make sure that all the fumes are being collected and neutralized. So there are some corrosive fumes being collected also by the scrubber. So here the scrubber unit is on the left side, and it's filtering and neutralizing all of these fumes. And yeah, once the measurement is done, I mean the digestion step, the lift moves to sample position automatically, and you can also place the drip tray there to avoid cross-contamination from dripping acid into your sample tubes. And afterwards, you just place the whole rack with all the samples into the autosampler, and you're ready for the next step. So the next step, as I mentioned, is alkalization and steam distillation. So this step is very simple, actually. You just add sodium hydroxide solution. And what you do with this addition is you just raise the pH so that the ammonium sulfate gets converted into volatile ammonia over here on the right side. And yeah, how this whole process looks in real life, you can see in this animation here. So it starts with the sample transfer from the autosampler unit to the distillation unit. So the distillation unit is the right one on the right hand side. And then you have the alkalization, so the sodium hydroxide is being added, and then at the same time the steam distillation starts. So hot steam is purged into your sample solution and collecting all the ammonia which is in the solution into this condenser element. So here it's condensing and slowly dripping into the titration vessel. So yes, and of course you heard already titration vessel. So, and this is also the next step. This is the titration and the simultaneous quantification of nitrogen. So you determine it via simple titration, which is a pH titration. So you can either use a pH meter or a pH indicator with a colorimetric sensor to automatically determine the content of ammonium in the solution. And yeah, this is being done automatically. And also, yeah, afterwards, the receiving vessel is being aspirated and cleaned, so you can start with the next run already. Okay, and now we talk about, I guess, the most interesting thing for you, which are the benefits. So, and I think there are quite some benefits to mention here. So first of all, it's a precise and primary method. The limit of detection, the LOD, is quite low with 0.008 milligrams of nitrogen. And so is the limit of quantification, the LOQ, with 0.02 milligrams of nitrogen, and the accuracy is quite high with more than 98% accuracy. And of course, IQ-OQ is available here, and we have an automated quantification via titration, which I showed you in the last video already. And if you use the autosampler, which you also saw in the video, you have a very time-efficient workflow, and you can really automate the whole process. And yeah, the best thing is yet to come. It is compliant to 21 CFR Part 11. And yeah, as you remember, this was the second learning goal of this webinar. And I guess it's a good point to move towards this 21 CFR Part 11 regulation here. And we're going to talk about the main aspects of 21 CFR Part 11. So here's a short overview. We just talk, about subpart A, which is mainly general provisions such as definitions. Subpart B deals with electronic records, and subpart C is about electronic signatures. So yeah, let's start with some definitions in subpart A. So in section 11.3, you distinguish between a closed system and open system. So a closed system is a system with limited user access to change items, whereas an open system is a system with expansive user access to change items. And also authentication is quite important. You either have a biometric authentication, which is using something specific to you yourself, such as a fingerprint or retina scan. On the other hand, you have a non-biometric authentication, which is possible, which is using something you know or have to determine your identity, such as a password or a smart card. So with our computerized system, we have a closed system and use a non-biometric authentication method, including a password. And yes, let's have a look at subpart B 11.10. This section is about controls for closed systems. And first of all, the invalid records, which is a good start to begin with. The system must be able to detect if invalid records were applicable, such as invalid field entries, fields left blank, fields that contain data values outside of the limits. Also, the system must be able to detect if the record was altered since its last approval. About viewing records, it must be possible to view and print the entire contents of the records. It must be possible to generate all of the records electronically in a format that can be put on a portable medium or transferred electronically. 1110C is about archived data. Data records must be protected against intentional or accidental modification or deletion. So, and now let's have a look how Büchi's computerized system is dealing with this section. About data records. So invalid field entries are recognized by the system, such as entries outside of the limits. Electronic records are saved and can be exported from the hard drive to a portable medium or via local network. And data records are protected from changes by any user. So you see, in summary, we can say we are compliant to 1110A, B, and C. So let's go to the next section, which is about access levels, 1110D. So the system must allow different levels of access based on user responsibilities, such as regular user administrator, if appropriate. So let's have a look about this feature of user administration here. So data access is restricted to unauthorized personnel and free user levels are available, like admin, lab manager, and operator. So the admin has access to all functions except the deletion of users. And he's able to delete and export and approve measurement results, but the system events log is completely trackable. So as I said, he got the permission to delete some files to make space on the memory for new results, but this will appear, of course, in the system events log as an entry that is, of course, completely trackable. And second is the lab manager. So the lab manager has access to modification of instrument methods and is able to export and approve measurement results. And yeah, eventually there's also the operator, which is just able to conduct measurement functions to generate results. And with these free user levels, we are compliant to section 1110D. Okay, so let's have a look at the next section, which is 1110E. It's about electronic audit trails, and electronic audit trails are really the key function of any computerized system which should achieve compliance to 21 CFR Part 11. So electronic audit trails must be automatically generated for the creation, modification, and deletion of records that are not required for non-essential automated background reportings, such as internal buffers, data swap files, and temporary files. So to say, they have to be generated for all quality relevant data. And electronic audit trails must be completely outside of the control and access of users except for read-only access of the audit trail file. Saying that you can basically, if you access the audit trail file, you're only able to read the entries and not change anything. And third, it must be impossible for the user to disable the audit trail function to make sure that there's a permanent tracking available. So let's have a look about this feature in our system. It's the data traceability. And yeah, the electronic audit trails are generated for all quality relevant data. The audit trail function itself runs permanently and cannot be disabled. And all entries in the audit trail are protected from changes by any user. And yeah, with these functions, it is compliant to 1110E. So let's have a look at some more main aspects. It's about electronic records and electronic signatures. So electronically signed records must contain the following information associated with the signing: the full name of the signer, the date stamp, timestamp, and the meaning of the signature. 1150G is about the authority to electronically sign. So the system must verify that an individual has the authority to electronically sign a record before allowing a person to do so. And we also talk about some electronic signature usage rules, which are electronic signatures must never be reused or reassigned to anyone other than the original owner. And the use of another's individual's electronic signature is prohibited even if the individual authorized such use. So it's not possible to borrow some signature for electronic records from another user. And now let's have a look about this whole section, how our system is performing in the feature of electronic signatures. So measurement methods and results are assigned automatically with the user information. That includes the name, the date stamp, and the timestamp, as well as the user level. And here you can see an example of how this looks like in the end. So on the top, you have the result. So you can see the date and timestamp when the result was obtained. And of course, also who created this result. So you see created by admin. So admin is the name in this case. And behind you see administrator. So administrator is the user level. And yeah, in the middle, you see then the blank value. So you also see when it was created, obtained. So you see the date stamp and the timestamp. And the method itself, you also see when it was created, let's say last modified. You have the date stamp, timestamp, and you see created by, again, the name and the user level over here. And then at the bottom, you see the approvement signature. You see approved by administrator, so the user level, admin, again, the name, date stamp, timestamp, and at the very bottom, which is quite practical if you have multiple devices and you have to distinguish between them, you see the serial number. So this is also, of course, always linked to the electronic record and included in the signature. So as I said, we provide traceable and secure signatures, and the signatures are implemented via a combination of username and password. So it's just the password from your account login. And the signatures are uniquely assigned to a user and cannot be reassigned to other users. For example, if you have a new user joining with the same name, they will not be reassigned to this new user. They will just stay with the existing one. And yeah, the signatures are linked to the respective electronic record. So whenever you export them, they are always linked to this record as well. So to sum this up, I hope you saw that the signatures cannot be falsified, and admins and lab managers are able to sign and prove results via the export function. So I would say compliance was achieved to 1150 and the section 1100. And yeah, let's move on to subpart C, also about electronic signatures and more about passwords. So password files must be encrypted or otherwise secured, so passwords cannot be read by ordinary means, including system administrators. And passwords must periodically expire and be revised. Yeah, this is important to make sure that they are changed on a regular basis. And section 11300D is about unauthorized use. So repeated attempts that are unauthorized via a non-biometric signature must result in an automatic disabling of the electronic signature. Yes, now let's have a look how we perform in this feature of login security. So combination of username and encrypted password is the only way to gain access in this system. Passwords expire after 90 days and have to be revised accordingly. So we make sure that they are changed on a regular basis. And yeah, the user account logout happens after 3 incorrect login attempts, just to make sure that nobody is trying, I don't know, 20 times with some random passwords to get access to the system. So yeah, overall, we are compliant to section 11300. And of course, we also offer IQ/OQ and training. So the validation of system is possible via IQ/OQ procedures and certificates. And I guess you're quite familiar with this topic, so I will not go into much detail here. And of course, user training is also available by Büchi. And all K375 documents are also available in electronic and print format according to ISO 9001 with the quality management system. So these were the relevant features for the main aspects of 21 CFR Part 11. So I hope I was able to give you a good overview about those features, and overall we are compliant to 21 CFR Part 11 with our computerized system. And as you remember, the third learning goal of this webinar was about 3 steps to establish a time-efficient and compliant SOP. So the first step is very quick and easy. Just check if your compounds in QC contain nitrogen atoms in their chemical structure. As I showed you in the beginning, it's a major part of all APIs, drugs also contains at least one nitrogen atom. So you can check this very easily, and we provide solutions as mentioned before, so digestion units and distillation units. The second step is more about also compliance to 21 CFR Part 11. So as you remember, I just mentioned all of these features, and they are packed into the pharma package firmware upgrade for the distillation unit. So you have the second step available is just to install the pharma package firmware upgrade, and you will have the compliance to 21 CFR Part 11. And last but not least, there's also a third step, which makes your life much easier and your work as well. It is about automation of your workflow for high throughput to save time and costs with our auto sampler function. So we have 2 auto sampler options available, one with 24 positions and one with 48 positions. So you can measure 48 samples in one single run. And yes, as promised in the beginning of the webinar, now we're at the end almost, and it's time for the bonus. So here now you will see a real-life case study of how Sanofi has adopted the Kjeldahl method in their laboratories. Okay, so let's first talk about the application. So what Sanofi is doing, they're using this equipment to determine the nitrogen content in raw materials such as polygylene and gelatin. So polygylene can be used as a stabilizer in vaccines, for instance, and they do quality testing of critical APIs, raw materials, excipients, and also drug products. And yeah, as you see, it's a wide field of application, which they also use. And the equipment that they use is the Büchi Kjelmaster K375 distillation unit with an integrated titrator. So this is the device that I showed you before. And yeah, their conclusion is Büchi instruments allow us to perform Kjeldahl method easily with reproducible and reliable results. We have previously used the B339 from Büchi. However, the K375 has many new fascinating features which allow fast and reliable analysis with maximum productivity. So yeah, I hope I could give you a rough overview of what they are doing and how they use the Kjeldahl method in their drug products and raw materials. So now we are already at the end, and I would say it's time for the Q&A session. So
Great, Eduard. Thank you very much for that. Now we got some questions prepared for the webinar and for the Q&A session. So let me show them. Okay, could you confirm that you see the presentation? And now, yeah, we can go with Q&A.
Yes, I can see it. Yeah.
Okay. So what I suggest, Edward, you could read those questions and right after just answer them. And the first question is.
Okay. I have a question. We purchased one Kjelmaster in 2015. Is it possible to upgrade to this pharma package in existing systems? Yes, it is possible. So it depends on the firmware version that you have. So you purchased in 2015, so the version of 1.5 requires a firmware upgrade, but all newer models, they really, you can directly upgrade to this pharma package.
Okay,
Yeah.
thank you very much. And we have another question.
Maybe I didn't completely understand, but I have a question regarding the audit trail. Is it somehow possible to delete it? Yes, so it is only possible to delete it if it is already exported. So it's very important, the hard drive, this will store a lot of audit trails. And yeah, if you never delete them, the hard drive will be full. So it is only possible to delete it once you export it. Yes.
Okay, thank you very much. Very detailed answer. And another one.
We have one API that we have in quality control. It is a pyrosol derivative, so it is an N-heterocycle. My question is it also possible to measure the N content in those aromatic ring structures? Yes, so aromatic ring structures, which include nitrogen atoms in their structure, this is just carbon and nitrogen bonds, and you can also crack them via the digestion process with sulfuric acid. So yes, it is possible to also measure N-heterocycles.
Good, good. Thank you very much. And another question.
What if there is some nitrogen impurity in the reagents, for instance, in the sulfuric acid in the digestion step? How do you take care with these interferences? So nitrogen impurity in the reagents, there shouldn't be any problem because you always measure blank values. So blank means you just measure the empty sample tubes to get a comparison to your result that you obtain in the end. So if there is an impurity, you also use the same reagents for the sample values and also for the blank values. So there is no interference. This is being canceled out.
Thank you very much, Edward. And another question is here.
As far as I understood, the sample transfer works via steam. I just wonder if the sample transfer is complete with this method. Yes, so it is working with steam, and you have basically the steam going through the auto sampler and then transferring your whole sample to the distillation unit. And this is being done throughout the whole steam distillation for many minutes. So in this case, you have a complete transfer of your sample, and in the end, even, I mean, the glass of the auto sampler will be completely clean. So you don't have to clean it anymore. It's a full transfer.
Great. That's great. We still have some questions, and that's another one.
Concerning the user management, how many users can be created on your system? So actually you can create as many users until the hard drive is full. So almost, I mean, it's not unlimited, but it's really a lot, like I would say more than 1,000 for sure. So, and yeah, you can, yeah, I think in practically manners, it's really unlimited. Yeah.
Good. Sounds like really flexible system. All right. Another question.
How does your system ensure that after login, no other unauthorized user is using the system? Yeah, once you have, yeah, I see. So once a user is logging in, then he's staying logged in, and if there is 10 minutes of inactivity, the system automatically logs out the user. So in this case, after 10 minutes of inactivity, no other user can use the system.
All right. Yeah. Okay, good. And another one.
I have a question regarding the sample throughput. We have approximately 40 samples each day. Is this possible to be measured on your system within a single day? Yes, if you choose the auto sampler configuration, so you have the auto samplers with up to 48 positions. And basically you can run your 40 samples within a single run. So you can measure them, for instance, overnight, and so yes, you can do it in a single day.
Okay, and another question is here.
One question about this measurement technique: what are the benefits in comparison to other techniques used in QC? That's a very good question. So as I mentioned before, the Kjeldahl method is a primary reference method, which means that you do not require actually any calibration, such as in spectroscopic methods. So basically it's an absolute method, and the results that you obtain, they are absolute. And second, I would say that it's highly selective. So it's very selective, of course, to all compounds that contain nitrogen. So you really make sure that no other compounds are interfering with your measurement.
Another one?
Maybe you mentioned it already, I'm not sure. Can you tell me how do lab managers approve and sign results? Yes, I mentioned it. It is about the export function. So whenever a file is being exported, it is automatically approved and signed at the same time. So just by exporting some data to another hard drive or local network, you basically approve and sign those results. Yeah.
Okay, let's see if we have another one. Yeah, we do. Yes, so another question is here.
How to calibrate the distillation unit from Büchi? So so the only thing you have to calibrate is basically the pH meter. So when you do a pH titration or the quantification, you need to calibrate the pH meter, yes, of course. And the distillation unit itself, you do not need to calibrate. The only thing that you have to do is to measure some blank samples, so blank values already before you measure your samples. So to make sure to know the blank value. Yeah, that's it.
Okay, thank you. And that's all we prepared for today. Thank you, Edward, very much for your presentation. Thank you very much, everybody, for your attendance and being with us during the webinar. I hope we will see each other soon on other big webinars. Stay tuned, and thank you very much for today.
Yeah, thanks a lot. Bye.
Bye.